In this video, the next functional group that we will be looking at are the carbohydrates. So the reason we care so much about carbohydrates is carbohydrates play a very key role in how we deal with energy and our metabolic pathways. So the key carbohydrate that's involved in this energy need and this metabolic pathway is glucose. And that's kind of one of our most simple carbohydrates. It's a simple sugar that all of our organisms need in some way to power their metabolism. So this has a key function in two different key metabolic pathways. First is if you go from left to right, the forward direction. So going from left to right, what you see is the process that plants are undergoing when they go through photosynthesis. Plants are taking carbon dioxide, water, energy from the sun, going through photosynthesis, converting those things into glucose and oxygen gas. Now, it's not this one-step process the way it looks here. There's actually probably 30 to 40 individual steps that happen. We're just showing the very beginning and the end, that what you start with and that very, very final end product. But you can see glucose is produced in this, and that's why it has such a key role in energy and metabolic pathways. If we think about this going in the reverse direction though, so kind of thinking about this going from right to left, what's happening when we go from right to left is cellular respiration. This is what every single one of our cells is doing in order to sustain life constantly. Every single one of our cells is taking glucose, reacting it with oxygen, going through that process of cellular respiration. That's how we're getting energy. Water gets produced as a byproduct, and then we also recognize that we exhale carbon dioxide as a waste product. So glucose has a key role. It's being formed by plants through photosynthesis. It's being consumed by every single one of our cells in cellular respiration. So glucose is this simple carbohydrate. It's really, really important. It's not only the end product of photosynthesis from plants, but it's also the starting point in cellular respiration. So it's critically important to our living organisms. So as we're looking at our carbohydrates, we're going to talk about three different types of carbohydrates. And we're going to build up from the smallest carbohydrates and then get bigger and bigger, and then finally finish with our largest carbohydrates. So our first type of carbohydrate we're going to talk about are the monosaccharides. These are the simple sugars, and these are the smallest carbohydrate units. Then we're going to see how monosaccharides can get linked together to form disaccharides. These would be two monosaccharides linked together. And then finally, remember, these biological molecules, some of them are very, very, very large. We're going to end with polysaccharides, which could be hundreds, if not thousands, of individual monosaccharides linked together. Polysaccharides is anytime you have more than two monosaccharides linked together. So that would be many monosaccharides linked together. So the first thing we want to do when we're talking about our monosaccharides is be able to classify our monosaccharides. And there's two different classification schemes. First, we want to talk about classifying them by functional groups. And in a little bit, we'll see some structures of some monosaccharides. But monosaccharides get differentiated into two big categories in terms of their functional groups. Is a monosaccharide with a ketone functional group, we're going to call a ketose. A monosaccharide with an aldehyde functional group, we're going to call an aldose. So that's one way we can classify monosaccharides. It's either ketoses or aldoses based upon which of those two functional groups they possess. There's another classification scheme we can also use for monosaccharides. That's based upon number of carbons. So instead of looking at functional groups, we could also just look at the number of carbons. A monosaccharide with 3 carbons would be called a triose, 4 carbons a tetrose, 5 carbons pentose, 6 carbons hexose, 7 carbons heptose, 8 carbons octose. And you can see those prefixes should be familiar to you. You've seen these before in other sorts of classification and other sorts of nomenclature. So we can look at functional groups and we can also look at number of carbons. What you see in terms of the ending for all of these is you see this ose ending. Aldose, ketose, octose, heptose, hexose, pentose, tetrose, triose, ose. Ose signifies that you're dealing with a monosaccharide usually. So think about some of the simple sugars that you're aware of. You're aware of glucose, fructose, sucrose, lactose, all have that ose ending that you're used to kind of seeing, that you associate with sugars. So when you see that ose, you're looking at, hey, that's a pretty good bet that you're going to be dealing with a sugar, a monosaccharide. And that's what monosaccharides are. It's a fancy term to really talk about sugars, and these are our simplest sugars. So here we want to look at some examples of some monosaccharides so we can practice classifying these. But first, before we do that, I want you to just look at these overall structures. And one thing you'll notice about monosaccharides is that the carbon chain in these monosaccharides is usually written vertically. Now with the organic compounds and some of the other things we were working on, you've noticed that we usually write the carbon chain out horizontally. So why are these written out vertically? Well, these are generally written out vertically because sugars have, these monosaccharides have something called chirality. And you can think about chirality as handedness. If you look at your left hand and your right hand, they are actually fundamentally different. They are not superimposable upon each other. So there's handedness in our hands. Our left hands are distinctly different from our right hands. These sugars also have that chirality or that handedness. Now that's not something we're going to talk about or go into any depth on, but that's the reason why these are written vertically, such that the handedness is a little bit easier to see. What we care about is being able to do our identification. So we're going to look at each one of these monosaccharides and then correctly identify them and classify them based upon aldoses or ketoses, as well as classifying them by number of carbon atoms. So if we look at the monosaccharide all the way on the left, first thing we want to pick out is it an aldose or is it a ketose? Now you'll notice in these monosaccharides you have a lot of OH groups present. You're always going to see a lot of OH groups present in these monosaccharides, so we're not using those to classify. Remember, we're picking out whether it's an aldehyde or a ketone. Well, we can see in this one we have a aldehyde functional group, so that's what makes this one on the left an aldose. Next, if we want to classify this based upon the number of carbon atoms, what would we have to do? Well, you just have to count the carbon atoms. 1, 2, 3, 4, 5, 6 carbon atoms. 6 carbon atoms would make this a hexose. Now if we go to the next molecule over, again, first thing we want to look at is this an aldehyde or a ketone. That'll determine if we're dealing with an aldose or a ketose. And here, circled, we can see we're dealing with a ketone. Being we have a ketone here, we are dealing with a ketose. So we have that checked off. We've identified the functional group that's going to classify our monosaccharides. Next, just like we did before, we can also classify based upon number of carbon atoms. So we just have to count the correct number of carbon atoms. 1, 2, 3, 4, 5. 5 carbon atoms would make this a pentose, pent being our prefix for 5. We go to the next monosaccharide over. Again, first thing we want to see is it an aldehyde or is it a ketone? This one is an aldehyde functional group. The C double bond O is on the end carbon. That's what makes this an aldehyde. Next, we would want to classify this based upon number of carbon atoms. So we just have to count how many carbon atoms we have. 1, 2, 3, 4. 4 carbon atoms would make this a tetrose. Now if we look at the monosaccharide all the way on the right hand side of the slide, here again our C double bond O is on an end carbon. That's what makes this an aldose. Then if we want to classify this based upon how many carbon atoms we have, we would just have to count out our carbon atoms. 1, 2, 3, 4, 5 carbon atoms. 5 carbon atoms would make this a pentose. So for classifying monosaccharides, it's just using things that you're already comfortable and familiar with. We're able to identify the differences between aldehydes and ketones. An aldehyde is going to make it an aldose. A ketone is going to make it a ketose. And we're very used to already from previous units counting carbon atoms. And that's all you need to do in order to successfully classify these monosaccharides. We want to look just really briefly at the structure of glucose, and we don't need to memorize anything on this in regards to the structure. This is more of a glucose being such a key carbohydrate in our metabolic pathway, photosynthesis for plants, cellular respiration for our cells, and we want to see what does the structure of glucose looks like. And there's actually 3 different forms of it. In the middle you see kind of the straight chain form. So here we see the straight chain form. There's also a couple of cyclic forms of glucose. And the cyclic forms of glucose result from kind of an internal reaction where the OH group kind of wraps around and closes in and forms a ring with that carbon of the aldehyde. So we don't need to worry about how that happens. That's just kind of how those rings get formed. And there's 2 different ways, kind of 2 different twists, 2 different directions that can happen. And that's what you see on the left-hand side and the right-hand side are these different cyclic structures of glucose based upon how that reaction actually happens. So even though we are used to looking at the open chain structure, it's the easiest one to pick things out. Most of the glucose in our body is in one of these cyclic structures. So probably about 99% of the glucose in our body is in one of these 2 cyclic structures, either on the left or the right. Less than 1% is in this open chain form you see in the middle. But we just wanted to show a quick diagram of what glucose looks like, being it's such an important monosaccharide for us. You can see it's an aldose. It has an aldehyde functional group, and if you count carbons, it has 6 carbons. So it's also a hexose. There's one key reaction that we want to do for our monosaccharides, and that's an oxidation reaction. So it's the oxidation of monosaccharides. This is sometimes called Benedict's test or using Benedict's reagent. Now there's some key things that happen in this. This is very, very selective. So this is a selective oxidation. What we're using to perform this oxidation are copper plus 2 ions. And that's very important. These copper plus 2 ions are only powerful enough to oxidize the aldehyde, leaving the alcohol functional groups alone. So we know both alcohols and aldehydes can get oxidized. We have a lot of alcohol groups in these monosaccharides. The copper ions cannot oxidize the alcohols. They can only oxidize aldehydes. So that makes it very selective. So this is a selective test for aldoses. Ketoses will not oxidize in this. And the reason this test gets used to try to identify aldoses is it's a very simple and straightforward test. It's also a very easy color changing test. You don't need fancy equipment or instruments in order to do this test. You can just look at what happens. In a positive test, what ends up happening is the copper plus 2 ions are blue. And what happens is if they undergo a change, what happens is they actually turn into a red solid. So it's a very quick and easy test in order to do to check for an aldose. So let's kind of look at what happens here in the molecule at the top as kind of an example of this test. First, if you look on the left-hand side, we see we have a monosaccharide written. And the key part that's going to change is the only part that's going to change is what I have boxed in purple is the aldehyde. No other parts of the molecule are going to change. All of the secondary alcohols below it, the primary alcohol that's also in the molecule, those are not going to change. So what happens when we perform this oxidation? Well, when we perform this oxidation, we know that aldehydes end up oxidizing to carboxylic acids. Again, you can see the change in the purple box. That's the only part of the molecule that's changing. So our aldehyde, just like we're used to, our aldehyde that we started with ended up getting oxidized to a carboxylic acid, just like we were used to seeing in previous units. The rest of the molecule remains unchanged. Now here's where the color changing part comes in. The color changing part is based upon what happens with the copper. Remember, the copper is what we use to oxidize this aldose. These copper plus 2 ions start as blue. So we can see that color very, very easily. Then, in the course of them performing that oxidation on the aldehyde, those copper ions also undergo a change. And that change that they undergo is that those copper plus 2 ions get changed to the Cu2O. And the Cu2O, this copper 1 oxide, this is a red solid. So you form a red solid, whereas before you had a blue solution. So that's that very quick color changing test that you can see. So this is sometimes called Benedict's test. Sometimes these copper plus 2 ions that are blue are called Benedict's reagent. It's just like the oxidations we've seen previously, except this one is a little bit more selective. The copper ions won't oxidize everything. It will only oxidize the aldehyde to the carboxylic acid. And as a result of that change, the change that the copper ions then have to go through, that gives you that very quick visual color change that you can use for a very quick test. So the next thing we want to do is start looking at how our monosaccharides can link together to form disaccharides. And the way these are linked together is that disaccharides are formed from combining of the OH groups from 2 monosaccharides. So this combining of those OH groups forms an ether-like linkage between the 2 monosaccharides and ends up producing water as a byproduct. So we'll see this with a couple of examples as well. And this is a reaction we haven't really seen before in terms of our organic chemistry unit. And the linkage typically happens from the OH groups on the first position of the first monosaccharide and the 4 position of the second monosaccharide. They're sometimes called 1,4 linkages. So I want to kind of show you how the different positions are identified in a monosaccharide. And for this, we usually look at these cyclic structures. So you can see an example of a cyclic structure of a monosaccharide on the right-hand part of the slide. And what you want to do is you want to look at the oxygen that forms the ring. Now that's not a carbon. We don't number things that aren't carbons usually. But that's kind of where we start, is that oxygen that's in the ring. And then we go clockwise counting the carbons. So looking at the oxygen, then we start going around clockwise. Here's our first position. Here's our second position, just going clockwise. Here's our third position, going clockwise. Fourth position, again, still just going clockwise. Fifth position, and then finally we can get that sixth carbon that exists outside the ring. So when you're looking at positions and trying to orient yourself when you're looking at these structures, you want to find the oxygen that's part of the ring and then just go around clockwise counting those carbons. So another thing we're going to look at is the geometric orientation of the OH groups with respect to each other, whether they're both up, both down, or one of each. What will happen with this is this will manifest itself in two different types of linkages in that product. You're going to see that the differences in these two forms is very subtle. But this subtle difference is critically important, so it's something we're going to have to pay special attention to. So let's look at an example of forming our disaccharides. So this one, just like any of the other reactions, we're only looking at key spots in the molecule where changes are happening. What functional groups are reacting? So to help us kind of focus in on where the changes are happening, what I'm going to do is I'm going to draw a kind of red box that shows both in the reactants and the products where all the changes are happening in. So all of the changes are happening in that red rectangle that I just drew. Everything outside of that red rectangle, you're not seeing any changes happen for that. So let's kind of look at what happens when we combine two monosaccharides to form a disaccharide. First, we're looking at the OH group from one monosaccharide in the one position. So that's what you see on the left that is highlighted in yellow. The OH group is on the one position. And then on the other monosaccharide, the one that I'm highlighting right now in yellow, is from the 4 position. So you see that's where we're getting the OHs from. Those are the ones that are going to react on the 1 position of one monosaccharide and the 4 position of another monosaccharide. And what's going to happen is those OHs are going to combine to form in the products an ether type linkage. Now notice what we started with. We started with 2 hydrogens and 2 oxygens. What did I have left? Well, I only have 1 oxygen here. Where did the other oxygen and the 2 hydrogens end up at? Well, you also end up producing water in this. And the water is not terribly exciting or interesting in this. We're looking at what are the changes that are happening in the biological molecule. But that's where those other pieces went. So here you can see we combine the OH from the 1 position with the OH on the 4 position to make that ether-like linkage. That ether type bond that we just formed when we're dealing with sugars is sometimes called a glycosidic bond. That's that ether-like bond. You can also see that it was the 1 and the 4 positions that we ended up using. So this is sometimes called a 1,4 glycosidic bond. Now the last part is kind of dealing with what's happening with the geometry. And to deal with that, we're looking at this is an alpha linkage. So what does that mean? Well, here, this is all based upon the geometry, how things are oriented in space. So in these alpha linkages, what you want to do is there's a couple different ways of identifying these. You can identify these based upon what's happening with the reactants, or if you're looking just at the product, you can also identify it that way as well. So I want to be able to talk you through how that works. So I erased some of my highlighting because we're going to relook at this time with respect to what's really happening with our geometry here. So what we want to notice, and I'm going to re-highlight this, is in the reactants. I'm re-highlighting these OH groups that we are using. I'm highlighting these in green. And what I want you to notice is that they are on the same side with respect to each other, meaning that they are both pointed down. So when you're looking at the reactants, if both of the OHs are pointing down or up, basically you have matching. So you're looking at the OHs, and in the two monosaccharides you're looking at, if the OH linkages are on the same side, so the same side could be, they could both be up or they could both be down. In this case, they're both down. Being they are on the same side, that's going to form an alpha linkage in our product. And now if I go back down to our product, and again, I'm going to highlight things in green. I'm going to kind of highlight this bond that's being made. And what we want to be able to see in this is a couple things. One, both of the rings are kind of still pointed in the same direction. Okay? They're both coming up from the oxygen. So that's your key thing that you're looking at in the product. There's another thing that you can kind of see even more easily than that in the product. And I'll show this both in the reactant and the product. And I'll highlight these in orange. So what I'm highlighting in orange up here in the reactants, at the end of those sticks, we often don't write them out, but they're hydrogens. And notice that those hydrogens are both sticking up on the same side. So they're on the exact same side. Being those hydrogens started on the same side in the reactants, they're going to stay on the same side in the products as well. And often that's the easiest thing to pick out when you're looking at a product, deciding what type of linkage it is. So when you're just looking at the product, maybe you didn't see the two monosaccharides that came together. You'd just be looking at the disaccharide on the bottom. The easiest way to identify what's going on with the disaccharide is to look at those hydrogens that I highlighted in orange. In your product, if your hydrogens are on the same side, so if our hydrogens are on the same side, here they're both pointed up. They could both be pointed down, but here they're pointed up. Being the hydrogens are on the same side, what you get is you end up getting what's called an alpha linkage. So in an alpha linkage, the OHs that you start with are on the same side in your monosaccharides, either both up or both down. The hydrogens, both in the reactant and the product, are either both up or both down. That's what gives you this alpha linkage. So this is an alpha 1,4 glycosidic bond. And what we're going to do is we're going to compare this to another type of geometry. We're going to see that on the next slide. So here we're dealing with what's called a beta linkage. Before we get into the geometry again, let's just kind of go through and show what's actually happening. Again, I'm kind of boxing in my red box. That's where our molecules are going to be changing at. That's the area of the molecules where the reaction is happening at. That's where we're going to draw our attention to. And again, before we even get into our geometry, we just want to look at what's happening. We're combining an OH group on the first carbon of one monosaccharide with an OH group on the 4 carbon of another monosaccharide. This in the bottom is going to form this ether type linkage. This ether type linkage is called a glycosidic bond. Just like we saw in the previous example, it still involves the 1 position and the 4 position. What's different about this one compared to what we just looked at for the alpha linkages are the geometry. The geometry, the orientation in space with respect to each other is a little bit different. So we want to point those out. First, we want to look at just the reactants and look at those OHs. So I'm going to erase my highlighting here just so we can kind of re-highlight that and re-emphasize that. Notice how in the reactants, the OHs are on opposite sides. Here you have one up and you have one down. So when we look at this, our OHs, we have one up and one down. So what we can say is when we have a beta linkage, our OHs are on opposite sides. Doesn't matter which one's up and which one's down. It's just that one has to be up, one has to be down. That's going to create a beta linkage. And that kind of gets maintained when we look at what's happening in the product. If I look down, you can see I have a very different geometry in my bottom structure than I had previously. Again, maybe the easiest thing to pick out as terms of whether we're dealing with alpha or beta is to look at the hydrogens. So again, I'm going to kind of highlight where these hydrogens would be in orange. Again, we usually don't write out the hydrogens, but they would be at the end of every one of these sticks. So here, notice in the reactant, the hydrogens are on opposite sides. In the product, that same geometry has to be maintained. These hydrogens are going to be on opposite sides. So if you're just looking at a product and you're trying to figure out, if you're just looking at the disaccharide and you're trying to figure out alpha versus beta, the easiest thing might be is to look at the hydrogens, because the hydrogens will quickly tell you the story. Notice how when we're dealing with the hydrogens are on opposite sides. One's up, one's down. So this creates a different geometry in terms of how this would actually look in 3 dimensions. So in the alpha linkages we talked about previously, you want to think about things on the same side, both up or both down, whether you're talking about the OHs or the hydrogens. For beta linkages, you always have things on opposite sides. The OHs are on opposite sides, highlighted in green here. The hydrogens are on opposite sides, highlighted in orange. And this difference is very, very subtle, especially when we're looking at it in a flat 2-dimensional drawing. But what we're going to see in a little bit is it's very important that we know the difference between an alpha linkage and a beta linkage, because alpha versus beta is going to mean an awful lot in terms of how these disaccharides and polysaccharides end up getting processed, broken down, and dealt with. So alpha, same side for OHs, same side for H's. Beta, opposite sides for OHs, opposite sides for your hydrogens. That's how you want to be able to distinguish and tell these apart. So now that we formed some disaccharides, we want to start talking about polysaccharides. Remember, these polysaccharides can be anywhere from 3 units linked together all the way up into the hundreds or thousands of units linked together. And what we want to do is we want to talk about some common polysaccharides that we're used to. The first types we're going to talk about are the plant starches. So these are the things that you would find in things like a potato. You would find these type of starches present, or in rice, or in grain, those type of plant-based starches. And there's 2 of them. The first one is amylose. And what amylose does is amylose is about 20% of plant starch. And amylose, all of the individual units get linked together in alpha linkages. So here we're thinking about alpha linkages. We just talked about differences between alpha and beta. These are alpha linkages. So not only are they alpha linkages, they're also all 1,4 linkages. So what that means is these will form a very straight chain structure. And you can see that with the diagram on the right. Because they're all 1,4 structures, you get very, very straight chain structures, like what I'm pointing at with that red arrow. They're linked together in a very straight chain linear fashion because all you get are 1,4 linkages. There's another type of polysaccharide that's found in these plant starches, and that's amylopectin. And amylopectin makes up about 80% of plant starches. Again, it's all alpha linkages, and they're mostly 1,4 linkages, but occasionally you're going to get something other than a 1,4 linkage. So every once in a while you'll get a 1,6 linkage. And what this 1,6 linkage does is it kind of stops things from being in a straight chain. It's no longer linear. Instead, now you're going to get these very highly branched structures. And you can see a cartoon-like level drawing of this. I'm pointing at it in purple. You can see you have these branch structures because it's not just 1,4 linkages anymore. You kind of get some 1,6 linkages sometimes happening. And what those 1,6 linkages do is they form branches. You can have branches coming off of branches, coming off of branches. You get this highly branched type structure. And you can see the differences if we actually draw out some actual structures all the way on the right hand side across the top. Because you are only getting 1,4 linkages, everything's going to be hooked together in a straight chain. But on the bottom, occasionally you're going to get a 1,6 linkage. I'm pointing at a 1,6 linkage in the bottom with the purple arrow. That 1,6 linkage ends up forming a branch. So for amylose and amylopectin, these are your typical plant starches. They're all alpha linkages. Amylose only has 1,4 linkages, so you get the straight chain structure. Meanwhile, amylopectin has occasionally you get a 1,6 linkage, so you get these highly branched structures occurring. We have a couple more common polysaccharides that we want to be familiar with and talk about. The first is glycogen. And glycogen is an animal starch. It's how, as animals, we end up storing our polysaccharides. So glycogen is an animal starch that is stored in the liver and muscles. And just like before, we're seeing alpha linkages. And for glycogen, they're mostly 1,4 linkages, but sometimes you get a 1,6 linkage. So remember, anytime you have something other than a 1,4 linkage, you're going to get these highly branched structures. So this is how animals store their polysaccharides for use as energy later. It's stored in our livers and our muscles as glycogen. There's one last type of polysaccharide we want to talk about, and that one's cellulose. And you're familiar with cellulose is because cellulose ends up forming the structural component of woods and plants. Think about bark, think about stems, think about those structural components. Cellulose is very different from the other polysaccharides we just talked about because cellulose is formed from beta linkages. So far, everything else has been alpha linkages. And they do form straight chain structures. And here's where that alpha versus beta difference is really, really important. Humans are not able to break down and digest these beta linkages because we lack the enzymes in our body in order to break down those linkages. So no matter how hungry you are, you could be literally starving to death. If you just tried to eat plant stems or bark from a tree, you're not able to extract the energy that's stored in there because that's in beta linkages. Your body couldn't break it down. If your body can't break it down, it can't use that later as an energy source. But we know some things do eat plants exclusively. Bark, stems, leaves. These are grazing animals. So when we think about grazing animals, things like cows, horses, those are domestic. We can have wild animals as well. Deer, rabbits, kangaroos if you want to get really exotic. They are able to break down these beta linkages. They can extract and break down these very long polysaccharides that have these beta linkages down into glucose units that they can use. And the reason being is not so much of what they have in terms of themselves, but their digestive tracts have bacteria that help them. It's those bacteria that have an enzyme that's needed to break down those beta linkages. So alpha versus beta linkages are very important. We as humans have the enzyme to break down those 1,4 linkages. So we can take amylose, amylopectin, glycogen, these very long, large polysaccharides with hundreds of monosaccharide units linked together, and we can break them down to get back those individual units because they're alpha linkages. We can't do that for cellulose because cellulose has the beta linkage. Our bodies do not have the enzymes necessary in order to break those down. Because they don't have the enzymes necessary to break those down, we can't ever access that. We kind of have to have that pass right through our systems without breaking down. We can't get at the energy that's stored in there. So even though the differences between the alpha linkages and the beta linkages are subtle in terms of the geometry, that very subtle and small difference means an awful lot in terms of what we can digest and process versus what we can't digest and what we can't process.